Hello everyone and welcome to my YouTube channel. Today in this video I am giving a demo of how I teach on the Filo platform, which I have been doing from the past 2 years, and I have taught more than 8,000 plus students on the Filo app, and I have also received many certificates from the Filo, which ranges from the gold, silver and bronze categories. So today I will focus on only giving the demo about the origin and the structure of the mitochondria. So you should know that mitochondria is an eukaryotic cell organelle, which means that it is present in every eukaryotic cells without any exceptions. And second, the function of mitochondria is to produce ATP during the aerobic respiration. Aerobic respiration means the respiration in which oxygen is required. Okay. It produces ATP during aerobic respiration. So it produces ATP, and ATP is called as the energy currency of the cell. So due to this, we also know that the thing which produces energy is also called as the powerhouse, because ATP is directly proportional to the power. So mitochondria is also called as the powerhouse of the cell. Okay. And aerobic means oxygen required. Okay. So, and third point is it is a semi-autonomous cell organelle. Only three organelles are semi-autonomous. One is mitochondria and other is chloroplast. So now let's move on to the origin of the mitochondria. The mitochondria is originated from the bacteria itself. So you can say that it is forming a symbiotic type of relationship inside our eukaryotic cell. Okay. Origin is due to endosymbiosis, or it is also called as endosymbiotic theory. Okay. Endosymbiotic hypothesis. In this hypothesis, there are two types of endosymbiosis are proposed. One is primary endosymbiosis and another is secondary endosymbiosis. In case of the primary endosymbiosis, a aerobic bacteria entered inside the proto-eukaryotic cell. Proto-eukaryotic means those cells which is between the boundary of prokaryote and eukaryote. Okay. So during that time aerobic bacteria invaded the protobacterial cells, okay, or proto-eukaryotic cells. And during secondary endosymbiosis, a cyanobacteria invaded the cell which is already having a aerobic bacteria or the mitochondria. So this formed mitochondria, and the secondary endosymbiosis result in the form of the formation of the chloroplast. Okay, so now we will see it with the diagrams. Sorry. So now what happened? This is a proto eukaryotic cell. Sorry if some noise come. I have tried my best to eliminate the noises. So this is a proto eukaryote cell. Okay. Now what happened? The primary endosymbiotic event is going to take place, and an aerobic bacteria invaginated. Okay. Or you can also say that this proto eukaryote cell just engulfed that aerobic bacteria. See, because this event is not seen by anybody, because the exact mechanism is not known, but it is proved that this is a bacterial origin thing. Okay, now I'll show you why. So this may be a aerobic bacteria, which this is a aerobic bacteria, and it is also proved that this aerobic bacteria can be rickettsia. Okay, modern day rickettsia can be related with this bacteria, which in the past time invaginated the proto-eukaryotic cell. So it entered the proto-eukaryote cell, and during entering, one membrane is provided by the host cell itself. Okay, proto-eukaryote cell, you can also call it the host cell, and the membrane of the bacteria formed the inner membrane. So this formed the mitochondria. So some cells did not took the secondary embiotic event, so they directly formed what? Fungi and animalia. Whereas some cells has undergone secondary embiotic event. And now in this case, a cyanobacteria, through the same way, either it invaginated the proto-eukaryote, or the proto-eukaryote itself engulfed this bacteria. Okay, this is a cyanobacteria. So again, due to its entering, one membrane is provided by the proto-eukaryote itself, and another membrane is of the bacterial origin. So these had both mitochondria and chloroplast, and these gave the origin of the modern day plants. Okay, so this is how the chloroplast and mitochondria both have originated. Now let's see the structure of the mitochondria. Sorry for that. This is how the mitochondria looks. The outer membrane, which is the eukaryotic origin, is regular in outline, and the inner membrane, which is the bacterial in origin, is having finger-like projection in the matrix, and these finger-like projections are called as crista. This is the matrix. The matrix is having 70 S ribosomes in it, and you know that the 70 S ribosomes are present in the bacteria, but it is also present in the mitochondria and chloroplast. And it also has its own genome, which is double stranded, circular, naked and histoneless, which is also the feature of the bacteria. It is double stranded, circular, naked DNA, which is histoneless, and that is why we say that the mitochondria is semi-autonomous, because it is having its own genome. But remember that the genome of the mitochondria is not sufficient to sustain all the activity of the mitochondria. It is dependent for some proteins on the nuclear genome. That is why it is called as semi-autonomous and not fully autonomous or completely autonomous. Okay, because it is dependent on nuclear genome for some of the proteins, and also the protein synthesizing machinery is all of the mitochondria itself. Okay, so the translation process or the transcription process is also similar to the prokaryotes inside the mitochondria or chloroplast. Okay, so I hope you liked my lecture, and if you want to contact me for any private lecture or any private tutoring, then you can contact me on my email. My email is singh@gmail.com. Thank you for watching this demo. If you have liked it, then please like and do subscribe. Thank you. Bye.